Okay, so this graph shows the importance of nutrient availability for bacterial growth. So this basically shows that as the nutrients become depleted, growth halts. So re-emphasizing the importance of nutrients. Okay, so there are several different ways to measure bacterial numbers. So the most obvious way is to directly count cells using a counting chamber. And this is easy, cheap, and quite quick. So the counting chamber is a microscope slide that has been specially designed with a grid to enable cell counting. So a drop of cell culture is placed on the slide, and then the scientist looks at the slide under a microscope and uses the grid to manually count the number of cells over a certain area. Then you also have automated ways to directly count the cells, for example, flow cytometers and electronic counters like the culture counter. But obviously these are much more expensive than the counting chamber. One of the major methods of direct cell counting is that it is difficult to distinguish between the live and dead cells. Then we also have ways of indirectly measuring cell numbers. For example, you can use viable counting methods such as the spread plate method that we referred to earlier. If you remember, you can grow the bacterial colonies, then the colonies can be counted, and then you can work out from the colony number how many live bacteria are in the sample. And then you can also measure cell mass. It's another indirect measurement, cell number. And you can do this through measuring the dry weight, the turbidity, or the amount of a cell component. Okay, so if scientists are carrying out a lot of experiments with bacterial cells over a long period of time, they will need an effective way to store bacteria without damaging the cells. There are several different long-term storage methods. The temperature which the bacteria are stored at affects how long they can be stored and remain viable. By viable, we mean living. So bacterial cultures can remain viable on agar plates at 4 degrees, which is fridge temperature, for approximately 4 to 6 weeks. And then in a supercooled freezer, so at minus 80 degrees, bacterial cells can remain viable for 1 to 10 years. Then freeze-dried bacteria can last for over 15 years at temperatures at or below 4 degrees. So the freeze-drying process is where bacteria are frozen and then placed under a strong vacuum so that the water sublimates, so it turns straight from ice into vapor. And with the exception of freeze-drying, the general rule is that the viable storage period of the bacteria increases as the temperature decreases. So we mean the lower the temperature, the longer you can store the bacteria for. It is important to know, however, that once the storage temperature gets below freezing point, cryoprotectants are essential to reduce cell damage caused by the freezing process. So commonly used cryoprotectants are glycerol and DMSO. Okay, so we've come to the end of the lecture, and I've just got a quick recap activity for you to do. Okay, so about 20 questions for you to answer to see how much you remember from the lecture. Okay, so question 1 is name 6 environmental factors that affect bacterial growth. And question 2, what is the difference between a psychrophile and a psychotroph? Question 3, acidophiles have their growth optimum in what pH range? Question 4, what is the name for bacteria that grow at optimal temperatures in the range of 20 and 45 degrees Celsius? So question 5 is what is the difference between a psychrophile and a psychotroph? Question 6, what is a facultative anaerobe? Question 7. If a bacterium is placed in a hypotonic solution, what happens? Does it A, shrivel because the cytoplasm contains less solid than the surrounding solution? Does it B, swell because the cytoplasm contains more solid than the surrounding solution? Does it C, shrivel because the cytoplasm contains more solid than the surrounding solution? Or does it D, swell because the cytoplasm contains less solid than the surrounding solution? And then question 8, if a bacterium is placed in a hypertonic solution, what happens? This is the hypertonic solution. So does it A, shrivel because the cytoplasm contains less solid than the solution? Does it B, swell because the cytoplasm contains more solid than the solution? Does it C, shrivel because the cytoplasm contains more solid than the solution? Or is it D, swell because the cytoplasm contains less solid than the solution? And question 9, what is budding? Question 10, what is the definition of a pure culture? Number 11, name 2 methods for obtaining a pure culture. And number 12, what are the disadvantages of using a continuous culture? 13, what is meant by batch culture? 14, how is oxygen removed from an anaerobic jar? 15, what is an anaerobic chamber? And 16, if you have a look at the test tube on the slide, which is the aerotolerant anaerobe in these thioglycolate broths? And number 17, what are the 4 phases of bacterial growth? Number 18, name 2 reasons why bacteria enter the stationary phase. 19, name 2 methods of directly counting bacteria. Last question, what needs to be added to bacterial cells when they are being stored at very low temperatures?